So, last time we were talking about hybridization and thinking about whether or not after hybrids form, they will be maintained or they'll be lost or they will change over time or they'll become stable. So you can think about this in a number of different ways. We start off here on the left with a population of organisms that are sharing genetic information as indicated by gene flow amongst those blue characters. At some point, if there is an event that subdivides the population and prevents sharing of gametes between individuals, and that could be geographic change, that could be a behavioral change. We talked about a number of different scenarios. Over time, those isolated populations will tend to become more different from one another. So these two populations can start to diverge. Initially, we might call these different populations subspecies, but if they become different enough to fail to be able to share gametes in the future, then those would become legitimate species in and of themselves. Somewhere along the way, though, probably when we're talking at the subspecies level, where organisms could share gametes, could reproduce successfully and make viable fertile offspring, usually there are some sorts of barriers preventing that from happening if speciation will continue to occur. But in those cases where these subspecies or closely related species can actually interact and share gametes, that's when we see hybridization. And as we said, some of these hybrids are fertile and vigorous, some of these hybrids aren't. So we talked about a number of different examples. One case was the case of reinforcement of reproductive barriers, looking at flycatchers with those populations of two species of flycatchers, the pied and collared, that lived in the same area. So those sympatric populations, the females were really good about selecting their own species as a mate. In the allopatric populations, that sort of barrier wasn't present, and females wouldn't do a good job of selecting their own species as mate if presented with a male from one species or the other. So in the case of those sympatric populations of flycatchers, there was behavioral reinforcement of that reproductive barrier, basically sexual selection. In the case of the cichlids, we saw a case where two relatively closely related species that can generate fertile offspring, can generate hybrids, seem to be doing so more often now because of changes in the quality of light making it through the water, and so less sexual selection is occurring. So in that particular case, because barriers to reproduction are being decreased, it seems that over time you might see loss of those two distinct species and fusion into that new hybrid species. And in a few hundred years, that hybrid species may be the only one left, and you might not see either of the parental species. And then finally, in the case of those two species of Bombina toads, the fire-bellied toad and the yellow-bellied toad, it seems like there's a relatively stable hybrid zone because the hybrids in that case aren't very viable. You can get offspring, but many of them have defects, many of them have problems during development, many of them just don't make it to adulthood at all. And so because those particular hybrids aren't vigorous offspring, they have very low fitness and they will tend to be lost over time. So in that particular case, we have a relatively stable hybrid zone that will probably persist over time, unless something about the viability and fitness of those offspring changes. All right, so chapter 26 is all about phylogeny, or the evolutionary history of organisms and the patterns of relatedness of those organisms. And this lecture should largely be review from lab. There will be a few additional thoughts and terms, but really this should all be very familiar to you based on the lab that you did this week. So the term phylogeny refers both to the concept that we're thinking about the evolutionary relationship of these organisms, but we can also call this tree, this phylogenetic tree, a phylogeny in and of itself. And we're going to talk about what the different branches mean and what the length of these branches mean and what branch points refer to. But at the end of the day, when you look at this tree, it gives you a good idea about our understanding of the relatedness of different kinds of organisms based on various features, whether those be genetic similarity or morphology, their shapes or anatomy, their internal structures. The science that uses phylogeny is called taxonomy or systematics. And in these disciplines, scientists use various kinds of features of organisms, and we'll talk about some of those features in more detail, to try to understand their relatedness and their evolutionary relationships. One sort of catch-all term you'll hear me use quite a bit is taxa or taxon, and taxa just refers to a group of organisms or individuals or types of organisms. So taxa could be a group of species. So in this case, we have geckos and snakes and iguana and monitor lizards and glass lizards. So you could refer to those taxa. But you could also refer to taxa that are sort of higher order on the evolutionary tree. So we could talk about plants and animals, for example, as two different taxa. So the term taxa or taxon, if you have just one, doesn't refer to any specific level of relatedness. It just refers to different groups of critters and can be used pretty generically when we're talking about phylogenies. So again, this particular representation is sometimes called a phylogeny or a phylogenetic tree or a cladogram. Any of those terms can be used to describe this sort of representation of relatedness of these organisms. And we'll talk about some of the features of these trees as time goes on. As you know from fall, typically when we're referring to individual species of organisms, we use a two-name or binomial system. And that binomial system is the genus and then the species of the organism in question. Note that the genus is always capitalized and italic, and the species name is always lowercase and italic. So in this particular case, we have two different species interacting. One of them is Homo sapiens. Sapiens or sapient refers to wise, essentially, and homo refers to man. So Homo sapiens is wise man, although in this particular case, I leave it to your own judgment. The other organism in this case is genus Felis and species Catus. Note that Felis catus is just the domestic cat, and there are wild cats of the Felis genus, and their species is Sylvestris, and they are very, very closely related to Felis catus. As a matter of fact, they very easily make fertile, viable offspring with Felis catus. However, there are behavioral and geographic barriers to reproduction of these two closely related species. So we still consider them distinct species because of those sort of ecological and geographical separations, even though they are the same biological species. So once again, the species concept and the labels we use are pretty imprecise and pretty messy much of the time. When we talk about genus and species, we're ultimately talking about a classification system that is called Linnaean taxonomy. So Linnaeus was a very early biologist in the 1700s who tried to describe the relatedness of organisms based on a variety of features. And ultimately, that's what we'll be talking about as we think about phylogeny. The particular characteristic of the Linnaean taxonomy, however, is that Linnaeus describes distinct and specific levels of relatedness, one of which is species, the next of which is genus. And This is a useful concept and allows us to think about organisms that are closely related species. So uh, the Felis genus, or Felis catus, the domestic house cat, is in the same genus, which is this next level of organization, as the wildcat, Felis silvestris, but they are in different genera than Panthera, which includes some larger cats, panthers, for example. You can imagine then we can go to higher orders or higher levels of Linnaean taxonomy. The next is family, Felidae, order, Carnivora, and of course carnivores include other things like dogs, bears, and those sorts of lineages. So now we're getting less and less related to one another, whereas within the family, all of those are various sorts of cats of one sort or another. The next highest order is class, so all of those organisms I've described. All the carnivores, for example, are mammals, but there are also mammals that are herbivores, so they would be in a different order than the carnivora. Phylum is a very high order, and all of the phyla, almost all of the phyla I'll be talking about today, are chordates. So the chordates that you interact with most are vertebrates, so that have a spinal column and a bony protective spine. But it turns out that there are other more primitive chordates like lancets, and we'll see an example of those, that don't actually have a backbone. They do have a spinal cord, and they do have a structure that becomes part of the spinal column called a notochord, but they don't actually have a backbone. We're all still considered chordates, though. The next highest level, which was the highest level when I was a kid, is the kingdom, and we're in the animal kingdom in this particular case, and there's a plant kingdom, and then there are prokaryotes, but it turns out that that's not a fantastic reflection of the evolutionary relationships, and so subsequently the highest level of organization has been described as the domains, and you've heard about the domains last semester. So eukaryotes are one domain, bacteria are another domain, and archaea are another domain. So the bacteria and archaea are both types of prokaryotes, and they used to be lumped into one kingdom. And the eukarya are all the eukaryotes that have nuclei. So that includes plants, animals, and other sorts of critters we'll talk about this semester. So when we then consider this Linnaean classification system and a phylogeny, you might think that these various branch points on phylogeny correspond to specific levels of Linnaean taxonomy, but that's not the case at all. The phylogeny branch points occur wherever differences occur between groups of organisms, and that doesn't necessarily correspond neatly to Linnaean levels of organization. So, for example, we have a number of species here from this leopard, a badger, an otter, a coyote, a gray wolf, and you see all of their two-part names or binomials. So scientists will often use the binomial, but we often use the common names as well. Just depends on the group you're interacting with and whether or not you need to make fine distinctions between closely related species. So that's the species level. On this particular figure, we see the genera, plural of genus, next. And note that in this top lineage for the leopard, we don't see a branch point just before or just after the Panthera genus. So in this particular lineage, that genus does not correspond to a branch point, whereas in the next level, the badger and the otter, you see that the genera are separated by a recent branch point. And then down on the bottom, Canis, basically various dog-like lineages, wolf-like lineages, occurs right before a branch point of coyote and gray wolf. So the branches in and of themselves do not necessarily correspond to specific levels of organization. However, notice as we go from the species on the far right towards the left-hand side of this phylogeny, towards the base or the root of the phylogeny, we're marching from the most specific level of taxonomy, species, back through genus, and then family, and then order. So as you continue your way back towards the root of these phylogenetic trees, you are going to higher and higher levels of taxonomic organization. But the individual branch points just don't correspond to any specific level of that Linnaean taxonomy. So there's a little bit of a disconnect there, but we just sort of roll with it. Okay, so let's look at this particular taxonomy of various reptiles. We have some geckos and snakes and lizards. If you look at all of these organisms, they have a variety of features that are shared. So, for example, all of these reptiles have scales that cover their bodies. And those scales are said to be homologous features because they are shared by this group of organisms due to their common ancestry. So all the way back on the left-hand side by the root of this tree, there's an ancestral lizard that had limbs, and presumably that had scales. So all of these descendant lineages also have scales. So scales are homologous features. And again, homology refers to a common ancestry. It turns out, though, that there's another way to arrive at similar features, and that's known as analogy. So if you look at the snakes and the eastern glass lizard, neither of these reptiles have legs. So initially you might suspect that they're closely related, but it turns out, as indicated on the diagram with the purple slashes, the loss of legs has occurred a couple different times in these various lineages. And the resulting similarity between snakes and the eastern glass lizard is not the result of those two having a common ancestor that was legless. This is, in fact, a product of what is known as convergent evolution, and we'll talk about this more over time. But in convergent evolution, one ends up with features that appear to be similar, but they've appeared independently through the course of evolution of those distinct lineages. So analogy is not because of shared ancestry, whereas homology is because of shared ancestry. So these features, like lack of legs in these reptiles, is called a homoplasy. That's an analogous feature, or one that has appeared independently over time and not through a shared ancestor. All right, so let's look at one of these trees and sort of talk about some of the parts so we can be on the same page when we're discussing levels of organization. As I mentioned before, at the left-hand side of this tree is the root end. So this is a rooted tree that has a beginning, and that number one corresponds to the common ancestor of all of the subsequent lineages to the right on the tree. So all of these taxa, all of these groups A through G, are related to one another through some ancestor at position 1. Not every tree is rooted. There are other trees that are derived in different ways, but this rooted tree is pretty common, and that's what you'll tend to see in this textbook. So if you consider the features that are shared amongst all of those taxa, you could sort of imagine what that original ancestor, that most recent common ancestor of all of them, might look like. At any of these numbered points where a horizontal line splits to a vertical line, so there's a bifurcation usually, goes from 1 to 2, that's called a branch point. Sometimes that's also called a node, and that indicates both a common ancestor, but also indicates two related groups that have evolved or diverged from that particular common ancestor. In some cases, it's not possible to understand which of three different groups is most closely related to one other. So in those cases, when you can't really decide which two of the three are most closely related, you might have a three-part branch point called a polytomy, but that's relatively unusual, and we generally try to avoid that. And often people will just sort of make their best stab at it to avoid generating polytomies, because those branches really should be two-part branches, not three or multi-part branches on these phylogenies, because we're really trying to understand the order of evolution of these different sort of organisms. Whenever one of these two-part branches occurs, each side of the branch is said to be a sister to the other. So in this particular case, B and C are called sister taxa. However, if you back up to that branch point number 3, you could say A is a sister of the group made of B and C. So you can define sister groups in different ways. It's not just sister species. There can be sisters at any level of the taxonomy. And then if you have a group that is clearly more distantly related to the others, or is perhaps the closest to the common ancestor, we'll call that a basal taxon. And in the lab, we used a basal taxon as our outgroup. So very often a basal taxon will be very, very important for understanding the relatedness of these groups of organisms, because that will help us make that outgroup. All right. Not only do we see changes in the structures or morphologies of organisms over time, but we actually see those changes as a result of changes in the genome or the genetic constitution of those organisms. So here we have DNA sequences from two very closely related individuals or species from some particular part of the genome. The genome is just the entire set of DNA of an organism. Over time, however, random changes occur in DNA sequences. So here in red, at the second position of the first sequence, you can see the C nucleotide has changed to an A nucleotide, and we can call that a substitution. So when you compare DNA sequences of closely related species, you'll see substitutions periodically. And as a matter of fact, you'll see substitutions within species as well. So in humans, about every 1 in 1,000 nucleotides is different between any pair of individuals, and those are the result of substitution mutations. Other sorts of changes include deletions, which are a loss of one or more nucleotides, or insertions, where one or more nucleotides are gained in a given sequence. So now, if we had a deletion of a G in the sequence number 1 and an insertion of a GTA in sequence number 2, when you stack up those sequences, now, after those first 4 nucleotides, they're no longer in sync because of the insertions and deletions. So there are particular algorithms or computer programs, computer methods, that have been developed to try to recognize the similar sequences and introduce gaps or spaces, placeholders if you will, in order to align those sequences that are derived from a common ancestor. So we'll call those homologous sequences. So by including the gaps, we can easily understand what areas of the original sequence has changed, and we can see the similarity in sequences when comparing any couple sequences. I should note that there are only 4 different bases in DNA: A's, T's, G's, and C's. So I would like to pose a thought question for you. If you consider any one position of a DNA sequence, what percent of the time would you have the same nucleotide at that position in any other random sequence? So it turns out that you can see substantial similarity in sequences even if you just start out with 2 random DNA sequences. There's a 1 in 4 or 25% chance that at any given position you'll have the same nucleotide in any 2 random sequences. So if sequences are truly homologous, derived from a common ancestor, then we would expect more than 25% conservation in the sequence identity or the sequence similarity between those 2 sequences. In distantly related organisms, that can be really hard to find. If it's right around 25% similarity, sometimes it's hard to tell if you only are examining DNA sequences from 2 organisms whether those are homologous sequences or not. So it can be very, very useful to have more recently related organisms to build a more complete comparison or more complete alignment to examine those sorts of DNA sequences.